Welcome everyone to today's ParaBoss webinar. Today we're going to be talking all about preventing theileriosis in Australian cattle herds. And today's presentation, as I just said, is brought to you by ParaBoss. For those of you who are new to ParaBoss, it's Australia's resource for controlling worms, lice, flies and ticks in sheep, cattle and goats. And it's a joint initiative from Australian Wool Innovation, Meat and Livestock Australia, the University of New England and Animal Health Australia. So today we have two leaders in the field online with us. We're going to have a presentation by Professor David Emery, and we'll introduce him in a moment. And we'll also be joined briefly to introduce the topic by Dr Matt Playford, who I'm sure you all know by now. We've had Matt on a few times. And he's also going to join David for a Q&A panel at the end of today's presentation. So I'll get to asking questions, but you'll be able to ask questions from both our presenters today. Also online today in the background, we have Megan Rogers, who is our ParaBoss Extension Lead. And Megan will be helping us run the webinar today. And for those of you who don't know me, my name is Fiona MacArthur, and I'm a member of the ParaBoss Extension team.
Yeah, thanks very much, Fiona. Just to get this disease in context, it's worth actually looking at where it started, and that's in Japan. And there's a particular tick over there called Haemaphysalis longicornis, which we call the bush tick, and in Japan they call Futatoge chimadani, and it's distributed over most of Japan. Now, where I was in Hokkaido, they had quite a bit of theileria disease in the cattle, but they didn't have any of these ticks. So it's obviously spread by a couple of other hosts, and they worked out there's a few other ticks that spread it. In the southern part of Japan, they got male and female ticks. In the middle of Japan, or the northern part of the main island of Honshu, only female ticks, and that means these ticks are able to complete their life cycle without sexual reproduction. So they do what's called parthenogenesis, and they're able to maintain their life cycle. Now, this makes them very distinctive, and the ticks that we're seeing in Australia are the same. You can collect hundreds and hundreds of ticks and not see a male, but you will see plenty of females. Okay, so that was the situation. Now, David's going to tell us a little bit about the other types of theileria, but the type of theileria we're interested in used to be called Theileria sargenti. It's the blue one. So it doesn't occur in Africa or Asia like the other theilerias, which are also very serious diseases. But it came to be sort of renamed Theileria orientalis. And about 2005, somehow it moved from Japan down to Australia, possibly via New Caledonia, but no one's really sure. But it arrived in Australia about then. 2009, we very kindly gave it to New Zealand, and then the disease continued to spread throughout Australia from that time. It's more recently appeared in the USA, and it's exactly the same strains and the same tick vector that's causing the problem in the eastern states of the USA. Now, looking at Australia, the disease first appeared in New South Wales, the mid-north coast of New South Wales. It spread south, it spread west, it spread north, until it actually was fairly common by about 2011. You can see all the blue dots there. Diagnoses of the disease, and the bigger the dot, the more places were positively diagnosed, and all the big dots are up there around the mid-north coast. And of course, it continued to spread when it hit Victoria, and this is thanks to Grant Rowland from Victorian DPI. It spread initially from the Murray Valley region, and then spread down to the Gippsland, and it's quite endemic in that area now, which means we get a lot of disease in that area. If we look at the distribution throughout Australia now, it's been diagnosed in Queensland, in South Australia. Some great research work done in Western Australia has identified it, particularly in that area around Denmark, in the southwest, and most recently in cattle that were actually bred in Tasmania, and it appeared in the clinical case there. So the Tasmanian department now are searching for ticks to see how they can better define that disease. So this is the up-to-date distribution, but David is going to tell us a lot about more recent research that he's been conducting. And just to introduce David, you'll be encouraged to know that David was born on a dairy farm in Gerringong. We can forgive him for that, but he loves livestock because of that. And through his career, he's focused on analysing the immune response to parasitic infections, identifying the pathogen or the causative agents that cause immunity or induce immunity, and formulating and delivering vaccines that contain these to provide a protective response for the animals. David did his vet degree at Sydney University and then followed that up with a PhD at Australian National University. From there, he studied Theileria parva, which is the African strain or species of Theileria in Kenyan cattle, and that's called East Coast fever. And then he joined the CSIRO Animal Health. Now, through the period between 1981 right up until 2000, when his lab was relocated to Armidale, David worked in the famous McMaster lab in Sydney and worked on several vaccines: trichomoniasis, foot rot, foot abscess, and the scourworm vaccines. After that, David joined Animal Biosecurity in Canberra, and then he took up his position as an academic researcher at the University of Sydney. And since 2012, when he was awarded his Doctor of Veterinary Science, he has been the chair of livestock immunoparasitology. David's also a fellow of the Australian Society for Parasitology. Since 2015, he has concentrated his research efforts on Australian theileriosis. And this has involved mentoring undergraduate and postgraduate students. And he's going to be giving us the fruits of those research projects in this presentation. So I'll hand over to you, David. Thank you.
Thank you very much, Matt. I'll just share my screen. So thank you very much. Look, it's a great privilege to be here, and I thank everybody for attending as well, and I hope you had a very nice Easter. Today, as Matt and Fiona have mentioned, I'm just going to provide updates around integrated parasite management for cattle, for controlling both the bush tick and theileriosis, hopefully. Now, it's called this horrible thing called BATOG, which is bovine anemia Theileria orientalis genotypes, which I promise I won't use again. So the structure of the presentation is really to transfer research findings over a number of years. And to do that, I just want to break it down into about 3 sections. Firstly, some basic facts about theileriosis to set the scene for management. Firstly, the theilerial life cycle, which as every parasitologist will know, is we are trying to break life cycles and break that into intergenerational transmission. A little bit about the final host, and I'll define that a bit later, and the vector, which is certainly in New South Wales, is Haemaphysalis longicornis. And a little bit more about the biology of transmission, because we try to avoid and reduce this. A little bit more about the disease in cattle and how the life cycle and that season relates to seasonality. How the disease progression moves through susceptible stock, because we're aiming to try and minimise the disease progression and getting animals to lose less productivity on the way. And just a little bit on the differences with cattle tick in Queensland and babesial tick fevers, because I would argue that Theileria orientalis is a little bit more difficult to manage. in terms of reducing the tick numbers. And then really to introduce the main thrust of it, and that's what we've found that could be used to reduce the transmission of the parasite, and how that can be actually put into an integrated management system for the susceptible populations, which are introduced stock and neonatal calves. My disclaimer down the bottom is here, I don't have all the answers. Like most of us, we're all a work in progress. And I'd certainly be quite keen to hear the discussion and to hear from your own experiences, because I suspect that quite a deal of you as producers have actually dealt with the carnage caused by this particular parasite, especially when your herds have been completely susceptible. So just a bit of background to this particular parasite. It has 11 genotypes, which I'd call strains or variants, very much like we classify COVID, right? So we had a alpha and a beta and a delta and an ipsa and an omicron and a B1, B2, and sound like bananas in pajamas. But it's very much the same thing. And because they've all got little bit of differences in their DNA, we can pick them up in these PCR tests, and you've also had your rat tests as well. And it's the same sort of thing. The more parasites there are, the higher the PCR goes up, but also your rat test, the colour you get on your test becomes more apparent the more virus you have, and in this case, the same with the more parasite. So the major ones that are problems over here, as Matt mentioned, are the ones that came in from Japan somehow. Chitosi and Ikeda are the two major ones that we have to deal with when we're dealing with disease. Buffalo is number 3 on the list, and it's benign, so-called benign. It's been in Queensland for 150 years, and we'll talk a little bit about why Queenslanders don't seem to get as much disease as we Southerners do, because they've had buffalo floating around there for quite some time. The other 8 genotypes are generally not virulent, although a couple of them can cause disease under circumstances. So the ones here, the buffalo, the Chitosi, the Ikeda, and the type 5 are in the current suite of assays that we have, our PCR assay, a bit low, so again, like COVID. And just down here, just a little reminder that bush tick doesn't really vector Theileria orientalis buffalo very effectively, which may be one reason why Queensland has a better cover against theileriosis from Ikeda and Chitosi, because buffalo is more better transferred by Haemaphysalis bancrofti, a different species of tick. Okay, a little bit about the life cycle, just to define some terms here. Because cattle are pretty big, we probably think, okay, well, that's the because it's big, it's going to be the final host, but that's not the case. The tick is actually the final host, and the final host being defined by the fact that sexual reproduction occurs in the final host. And for Theileria, that actually maintains its virulence. It's the poor old cattle over here, just like us with malaria. The poor old cattle are the intermediate hosts in this, what we call an indirect life cycle. So the intermediate host suffers all the pathology, unfortunately, and that's where the asexual replication occurs. And that asexual replication divides in the red cells. And every time it replicates, it breaks out of the red cell, and then it invades 2 more, and so on and so forth in a factor of 2. And so after a while, you start to get your red cells depleted, and that's where all the symptoms occur. So the poor old intermediate host of cattle in this particular case are the ones that suffer the symptoms, and the tick gets away relatively scot-free. Okay, bush tick, Haemaphysalis longicornis, which we did show was the vector in New South Wales. It's a 3-host tick, right? Which actually means that the eggs there in the grass, and then the larvae hatch out, they hop onto host number 1, feed, drop off into the environment again after about a week, and molt to the nymphal stage, which then grabs the second host, feeds, engorges, falls off, and molts in the environment again to the adult stage, and the adult picks up a third host and feeds the female, dropping off to lay the eggs in the environment. Now that's different to the cattle tick, Queensland cattle tick, which is now called Rhipicephalus, and it lives on the animal for around 4 to 5 weeks, feeding. So at all the stages actually mature on the animal. The female falls off, lays the eggs in the environment. So it's a one-host tick. So that means it's probably easier to control because the tick's all on the host, either that or it's in the environment. And the environment actually has the larval ticks, which are probably the most susceptible to pasture spelling and to heat control when we're dealing with the various stages of the parasite. So the larvae are probably the most susceptible stage. But in order to get from one host to another to transmit babesia and tick fevers, it's got to go through the egg, because the rest of the cycle's on here and you can't get to another host. The best way is to go through the egg, hatch out. A whole proportion of the larvae are then infected and can transfer the infection to whatever cattle they pop onto. Okay, but Haemophilus longicornis, in being a 3-host tick, is again not like the Queensland one, which only lives on cattle. This one actually can complete its life cycle on a variety of different species, a variety of different hosts. So we certainly ran it on dogs, and it can go through all the stages on the dog. It has other hosts too, kangaroos. We don't know too much about other sorts of macropods. But the interesting thing about this is it'll feed about 3 to 7 days at each particular stage of the life cycle, then it drops off. And when we actually see the clinical signs, which we'll get to, it explains some way why some diseased cattle may not have too many ticks sitting on them at any particular stage. So keep in mind that this thing can actually have a number of different hosts, which makes a little bit of a complication to its control on pasture. So what does it all mean? Well, it means that if the infection can't go through the egg, it has to be picked up new each generation. So obviously the larvae or the nymphs can pick up the infection from an infected cow, and then after the molt can reinfect at its next stage. So only the nymphs and the adults can carry the infection because it doesn't go through the egg. That's the first thing that we do about those. The second is that we found that even if they get infected as a larvae here, they don't need to reinforce that infection as a nymph. They can carry it through 2 molts and actually infect as the adult stage. So even if it feeds off kangaroos here, it can still carry the theileriosis infection and infect cattle as the adult stage. So therefore, this is a little bit of a complication in terms of how we actually control this. And we also noted that theileria takes around 3 days to mature the sporozoites before they're inoculated, before it's infected. And this is probably because if you have a look at a molted nymph or a molted adult, that there's not very much inside. And so they need those feed and the protein meal there to mature the sporozoites before they're inoculated. Which gets back to a very important thing about protozoal parasites and their transmission. If your treatments can then kill the ticks in 24 to 48 hours, then you can actually prevent the transmission and infection of Theileria orientalis. This is very much like what occurs with paralysis tick as well, which takes where the female takes, or the tick takes around 3 days to really ramp up the toxin production. If you can kill them in this early period, you won't get the transmission, you won't get the signs of tick paralysis as well. But keep in mind for Theileria, if you can kill them early, then you'll be able to prevent transmission of infection. So that's an important point, because most of our acaricides that we use nowadays will kill ticks within that period. Okay, so what's it all mean? The first thing is that the life cycle does lead to the seasonality. So you have this large sort of group over here in the right-hand side. The seasonality is also dependent, of course, on the provision of a susceptible group of animals, either introductions, but obviously with the spring, the birth of calves. who aren't immune to this particular parasite. When the epidemic curve went through, we also saw a peak in the autumn as well. But most of our enterprises now in the endemic areas actually have the seasonality peaking in the spring. So, but keep in mind that in endemic zones, you have ticks all year round, okay? Don't be deceived by the fact that no cases means no ticks, because they're certainly around. It just means that all the rest of your cattle have actually gone through the first peak of parasitemia that we'll talk about in a moment, and they've reduced their parasite load and become carriers. And those carriers are resistant to seasonal tick infection unless you really, really stress them. Okay, so you're waiting, basically, everything gets quiet on the enterprise through the winter time until you have some introductions or you provide a new susceptible group like calves. So that's because all your resident cattle in your endemic areas are carriers. We'll have a look at that in a moment, what it means. But the usual seasonality of ticks is, okay, the eggs are around in January or so, and then you have your larvae in the autumn. Your nymphs are the ones that go over the winter time, and then your adult ticks appear in the spring. Okay, the second component of it is that the nymphs do overwinter in the field. The interesting thing is they do stay infected for all of that period, very much like snails with liver fluke. They stay infected for up to six months, because we've tested them. And obviously, if they're able to over the winter, they're very hard to kill off with pasture spelling. Okay, they're pretty resistant. They also need some warmth to move, so that's another thing. And remember that bush ticks can carry Theileria through a kangaroo as a second host and remain infective. So that actually means that if you're removing cattle from your paddock with the idea of spelling it and trying to get the tick numbers down, and there's lots of roos grazing around there, you are only getting it reinfected. Okay, just keep that in mind. This parasite is all about the anemia. So here we have a little parasite, not a great one, a theileria stuck inside a red cell here, and you can imagine that when this divides, it breaks open the red cell, infects two more, and so on and so forth up the line, until you start to lose the number of red cells, and then you get this thing called anemia. You don't have enough red cells for enough gas, and the parasite, as the parasite divides, so you get all the rest of these symptoms more or less follow from the fact that you're losing your red cells. So all the fever, the lethargy, the production loss, and then finally death, which is a lot less common with Theileria orientalis than I witnessed with Theileria parva overseas in Kenya. It's a very virulent parasite compared to this one. Okay, so for transmission, you need to keep in mind that basically as soon as an animal hits the ground in an endemic area, it's infested immediately by ticks and infected about 3 days later. How do we know this? Okay, I'm going to go through this fairly slowly. The y-axis up here is increasing parasite levels. All right, this is like your results from your rat test or your COVID PCR. So the higher the dots here, the more parasites you have, as determined by the number of copies of its DNA. And what we did is we compared the parasites in the PCR test. So we've got over here a comparison between Ikeda, which is a virulent variant, a strain, versus the buffalo, which is the benign one in Queensland. So we looked at the age of the calves, and how we did this was we just bled them once. And since the calves have a sort of an 8-week calving period, which was from the joining period, obviously we're getting a range in the ages of them. And these were done at Dorrigo in 2017 and 19, so they're August calvings. And you can see 100% of calves, virtually 100% are positive for Ikeda by 5 weeks of age. You can see the peak is around here about between 5 and 8 weeks of age, which meant they had to get infected down here pretty quickly from the kinetics that we saw, because we compared them with introductions as well, where we could get more bleeds. Now, the infection slowly decreases with age. So you see by about 4 to 5 months here is getting right down to low levels. Compare that to buffalo over here, and two things stick out pretty quickly. Firstly, only 40% are positive for buffalo by about 5 weeks of age, and it slowly increases. And that's pretty characteristic of the difference between a virulent strain and a benign strain. And the other difference is, if we look up here, here's 250 up here and 70 there. So the buffalo parasite numbers are about 6 to 7 times lower than those for Ikeda and Chitosi, the virulent ones. And what you actually notice here over time in both calves and introduced stock is that these levels fall for the virulent ones down to basically what the benign buffalo does all the way along. And that is characteristic of the levels of parasites we see in carrier cattle, right? And that's what happens after they actually recover from that first wave. They go down to carriers at a very low level of parasite load. Okay, and that's shown again here. And these are arrivals or their calves. They show exactly the same sort of pattern under heavy tick challenge. And this is the death toll number, around about 3,000 up here. We don't need to worry about the particular level, except that, you know, introductions, up it goes, and we're sort of getting high numbers up here. And it's set around this 6 to 8 week period appears to be the maximum parasite load that they get. And after that, they come down here, and you can see buffalo along the bottom here has very low numbers. And this is a particular characteristic of a carrier state, right? By about 3 months, you can see that all the parasite load is quite low. It's subclinical. We're not seeing any disease. But this interesting situation is these carriers. We've taken them out for 30 months. They seem to be probably likely carriers for life. Now, along with this is the red cell numbers. You can see the red cell numbers are starting to go down around about the time of the peak parasite, because it's blasting out of the red cells and infecting more and blasting more. And so your numbers of red cells are going down. But, of course, as soon as you pass this peak of parasitemia, the PCV and the red cells start to increase back to normal levels. And the same with productivity. As I said, it's likely in the end that all cattle in endemic zones are carriers, as we shall see. So the carrier's state seems to be able to protect them against seasonal rechallenge unless you really stress the animal. Like you put a pregnant animal in about the seventh month of gestation, stick it in the back of a cattle truck and take it for about 200 to 300 kilometres over the ranges and somewhere else. We noticed when we're moving cattle to Dorigo that if they're not pregnant, steers that are carriers, we can transport them 700 kilometres from Mudgee to Dorigo without any sort of problems whatsoever. So the whole thing about control of Theileria is really getting them through this first peak of parasitemia in the first 6 to 9 months. First load of parasites they get, because everything gets steady state after that, when they're recovered. So what are our options for control? Well, firstly, I just want to recount a couple of things and provide you a little bit of evidence. The endemicity, virtually every tick and bloods we've collected from Dorigo, Milton, Stroud, Cabargo are all positive for all the Theileria strains that we've got in our kit to test. So therefore, it's virtually a round robin of ticks infecting on multiply infected cattle, becoming multiply infected themselves, and then transmitting that into the next susceptible population. We do know that between 6 and 50 infected bush ticks from the Tick Fever Centre will induce PCR positivity about 4 weeks after infection, or in 10 days or so, but we don't know how much is generating those rapid peaks at around 6 weeks of age, how many ticks are on there. But obviously it's a moving feast, because ticks are attaching and falling off, and more ticks are attaching and falling off as they complete their life cycle. So it's a real dynamic in an endemic zone. The other thing is there's no vaccine so far. For Theileria orientalis, it's not viable because the market's so small, and so none of the commercial companies are going to be interested in it. We may keep an eye on the USA. If it gets really bad over there, they might put a few bucks towards developing a vaccine. Buparvaquone is effective at reducing parasite numbers. It's effective against Theileria parva as well, but it's not registered in Australia. But the other thing about buparvaquone is it will reduce the parasite number, but it will not remove all the parasites. So it creates the carrier state. And I would argue very strongly that the carrier state is a great state to be in, because basically you're protected against re-challenge by the tick. And if it's seasonal re-challenge, it'll probably boost immunity, whatever that immunity tends to be. We don't quite know exactly what that is for Theileria orientalis yet. Okay, so our options, we are trying to slow down this rate of infection, so we haven't got this such a peak early on, by reducing the tick challenge. So the parasite levels rise more slowly, a bit like buffalo eye, and then two things happen. One is that the peak parasite level is lower, so we get less sickness, less disease, less production loss. And secondly, the animal, because of this, has a longer time to gain immunity to the parasite, to maintain red cell numbers, and therefore suffer less disease and be still able to feed. Good nutrition certainly helps. As we all know, since we haven't got we haven't got the buparvaquone, we're restricted pretty much to tender loving care and nursing of our animals. We'll actually see the majority of them get through that first peak of parasitemia. Once the animal gets into the carrier state at around about 3 months, it's protected. And that's sort of a bit of a godsend, if you like, for this particular parasite. And then some of the weight loss that we actually get may be regained by compensatory gain if the nutrition is good. So what do we know that can actually help achieve this? Well, firstly, I just want to provide a little bit of evidence that gives it some confidence that I'm not just talking through my hat, because I can't talk through my hair because I haven't got much of it. Okay, so parasitosis is lower and slower in cooler months. And this is the normal, right? This is the normal we get under heavy parasite load when there's plenty of ticks about. We get this peak around about six months. Right? We want to keep it below this, where we start to see real clinical disease and deaths start to occur. So if we send up cattle in February or September to Dorigo, all cattle will be infected in the sort of normal peak of around about 6 weeks. Okay, the maximum parasite load will reach about this high, but unfortunately, in these particular cases that we ran, we didn't see any deaths, but we did get about a 5 to 20 kilo weight loss due to over this particular period here. Now, in the wintertime, we couldn't regain that because the nutrition wasn't good enough. But in the September cattle, when they came in, they had sufficiently good nutrition during the summer and the autumn months to regain that 20 kilo loss by compensatory gain. The other piece of evidence was that we had cattle going up there in early May last year, and it completely sort of wrecked my experiment, but it did provide the evidence that I'm looking for here. What happened was that the maximum temperatures up there at Dorigo were 2 to 3 degrees lower from June to September because we had a really cold spring, and the tick infection rate was a lot slower. The cattle weren't all infected around this 6-week period, but it took another 6 weeks for all of them to be infected. And when we looked at the parasite levels, wow, the parasite levels were much, much lower when this sort of pattern occurred, this sort of level of infestation. So we're able to drag it out, and the parasite levels were up to 1,000-fold lower than what we anticipated if we had it all around here under a normal season at 6 weeks. We didn't get any deaths, and we didn't get any significant weight loss either, which wasn't what I was looking for, but it certainly serves to illustrate the fact that if you can slow this infection down, you can actually get animals suffering a lot less. My salutes to and salutations to for down at Milton, and where we actually had calves that were born there, and Mel Shea did a fantastic job of getting around these, patting them on the head, popping a python tag in around about 1 or 2 days after birth. And you can see here that had the effect of actually slowing down the level of parasitosis. Even after 8 weeks or so, it was still lower, getting dragged out. And even though it was miserably wet and rainy down there in the season last spring, there were very few, I think, very few losses attributed to Theileria, mainly problems with pneumonia, anything else. Dr Max was given at 7 weeks. 7 weeks is too late, because we saw back there in the earlier slides that the peak of the first peak of parasites is around about 6 weeks after infection. So I must admit that both of these, both the Maxima and the Dr Max, are off-label, which means that the company that manufactured them is not responsible for the outcome. You are, or I am. So this is an off-label treatment, but it seems to work. Dr Max is actually available for treatment of neonatal calves, so therefore it could be used there. But keep in mind that this is not a label claim by either of these manufacturers. Now, last year, Matt gave you a list of treatments for bush ticks on cattle, and I just refer back to those at this stage, because Matt will be available for a bit of a follow-up on that, because he knows more about this particular area than I do. Okay, so what options have we got? If we look at the environment in terms of trying to spell pasture like they do in Queensland for Queensland cattle tick, it's just not as easy. One, we don't have the hot summers. Our coastal epidemic areas are really quite suitable for the survival of ticks, because they get a bit more rain and they're much more amenable to the survival of the tick. And I would say that unless it's cleaned in autumn, winter spelling won't remove nymphal ticks. They are quite resilient and they remain infected for the whole period over winter as well. Summer spelling and grazing may be effective if you can graze it down, because if you treat cattle 3 days beforehand or so to kill off the ticks, and they're removed at the end of February after they've grazed it all down, it's a system very like smart grazing is for sheep worm parasites. Pastures, need to keep in mind too that pastures may be consistently infected if there are roos about, and I've up and down the coast, there's plenty of kangaroos, and you can't do much about those. You can't shoot them off or anything like that. It's not possible to control them without a heck of a lot of bureaucracy. And I certainly don't know about wombats or possums as well. Certainly we gave the possums to New Zealand as well as Theileria orientalis. In terms of calving times and sites and timing of introductions, a number of these things have actually been worked out by producers up around the Dorrigo area, because they've had so much sort of problem with it that certainly the first recommendation was to try and carve away from paddocks which have been infested with roos. In other words, stay away from the bush or roo-infested paddocks to try and reduce the immediate tick infestations of the newborn calves when they're just secreted in the bush, for example. The other thing is to try and carve a little earlier in the season when the tick is less active, and they've moved from August to July, where infections are likely to be slower, as we showed in the slide with the cooler temperatures. So introduce cattle in the colder months, if possible. Maybe keep them a bit close to the house paddock, because the colder months, obviously, those ticks are less active and we're going to get a lower infection rate. As I said, we just need to get them past that first peak of parasitemia, first peak of parasites. And so even just a treatment at a couple of months apart would be sufficient to get through past that first peak under normal circumstances. So the hosts and the tick numbers, the tick treatments, I'm going to refer back to Matt for that, because he gave you a good rendition of that in 2022. Keeping in mind, of course, that stock are infected almost immediately. So for introduced stock, you probably need to treat them before they arrive, and then regularly, just a couple of months maybe enough to be able to get them past that first peak and get them into the carrier state. That's basically where we need to get to in endemic zones. So calves from the previous slide on sites and timing, but I just admit straight off that more work needs to be done to look at trying to protect calves, because it's not as easy by any treatment that we use. But I would implore you to sort of think about your management, about how you might actually be able to treat them as soon as possible after birth, again, to reduce the tick challenge and slow down the rate of acquisition of the infection. As I mentioned, Dectomax injection can be used in day-old calves. It's active for at least 28 days against the Queensland cattle tick, so I would expect it would have a residual effect, a persistence period of something similar for bush tick. But two things you need to look at. One is you probably only need a couple of treatments at monthly intervals, but you need to watch the weight gains of calves over this persistence period to make sure that by the time they get to the end of it, they're not at too low a dose rate for the compound to have an effect. Okay, Matt knows more about this, and I'll sign over to him in a moment. But I thank you very much for this opportunity to sort of update you, and we may have some more material a little bit further down the track.
Thanks, David, for the presentation. That was great. And yes, I'd like to invite Matt to turn his, and we could leave that slide up actually that you've got up there right at the moment, because I have a question on it. But I'd like to invite Matt to unmute himself and get online with us again.
I'm unmuted now. Thanks, Fiona.
Hi, Matt. Thank you.
And now let me just say thank you very much, David. That was really fascinating and gave us a lot of important new insights into how to stop this disease. And as Fiona said, we've had a couple of questions, and the first one relates to treatments.
Since it's your slide, Matt, if you could talk a little bit more about the off-label versus on-label, please.
Yeah, so the products that are written there in green, you can go down to your local rural store and pick them up, and you can use them, no worries at all. So that's what's called a label dose. They've got a claim against bush ticks, and we know that they work against bush ticks for the particular amount of time that it claims, because the trials have been done. Now there's other products, the ones in orange or that orangey pink colour there, that we've got a fair bit of information about them and the way that they work, because they work on other ticks like cattle tick, as David mentioned, and he's actually done trials. They weren't registration trials, so they can't go on the label, but at least David's satisfied their research requirements that these products probably work to keep ticks off cattle, particularly young calves, and the Doramectin, the Dectomax injection worked on young calves because it's registered for use in any age animal. And then the ones in red, and now these are the new chemicals. Now you might be familiar with them because they're used in Simparica, Credelio, Bravecto, and NexGard that people use on their dogs, and they've actually got a claim against bush ticks in dogs, and so has Advantix, just by the by. Now they can't be used in livestock because they don't have any residues data, and because livestock are food producing animals, it's very important that we protect our trade by only using products that are registered. So just to go through them in a bit more detail, the first one is the synthetic pyrethroids, and the best example is Betacol spray-on. Note that we can't use Betacol pour-on because it's only for use in overseas markets, not to be used within Australia. But the Betacol spray-on is quite quite a good product to choose because it's got, low side effects and impacts on animals, and it's fairly easily applied. Same goes for arrest, fly and tick, dip and spray. The other one that's a dip and spray is this one, Barricade S. It's actually got a knockdown claim and up to 14 days protection, so it's quite a handy product. Only warning that you might say is that because it contains a organophosphate chemical, then it's probably a little bit, requires a little bit more care when you apply it in terms of both safety to animals and to people. Another product that's used very widely in Queensland is Amatraz, and again, that's available in a dip and spray. It doesn't give you any length of protection though, it's just a knockdown. So it's basically good for clearing animals, but won't give you any length of protection. And then of course, as David mentioned, products like Dectomax injection, they don't have a claim for bush tick, so it's an off-label use. Legally, you're required to get a vet script to use these products for bush tick, but we do know that they have good safety profile, and in the research field at least, they've been effective.
We have a few more specific questions about actual treatments, Matt and David. So we'll just go through some of them now. And the first one is from a producer down in Tasmania. Oh, sorry, an advisor. I advise producers on what bush tick treatments to use on animals imported into Tasmania. What treatments for which species would you recommend?
Yeah, thanks, Bruce. Well, look, any of these registered treatments are probably your go-to, given that bringing animals into Tasmania at the moment, it's a very delicate issue because Tasmania has only had the one case, and also bush ticks haven't been identified yet, to my knowledge, and so any introduction could cause a spill-on effect. I'm not going to say a disaster, but there could be more serious consequences in other areas. So any of these registered treatments are probably going to give you a pretty good result. The other cautionary thing that I'd say would be, a lot of people are buying cattle and shipping them around the place, and if you don't know the status of the cattle, then you can request a blood test to be done, which will quickly identify if those animals are carriers of the disease.
Thanks, Matt. And that was the second question by Bruce there. If you want to determine whether the bush tick is present on your property, what techniques would you use? And is it just the blood test there, Matt, or is there anything else that you would like to add?
Well, I think Bruce is specifically asking about bush ticks there. Now, bush ticks are problematic because they don't go questing like paralysis ticks do. You can't pick them up on a blanket. The easiest place to find bush ticks is actually on the stock themselves, and you can actually have a program where you go and feel the cattle up and down their legs and the back end, up around the udder and under the tail, and on their brisket, because they're the most likely places to find them when they first hop on.
Yeah.
Thanks, Matt. Next one, quite a few reports of producers using Baycox oral at induction, off-label, acknowledged, but could it be helpful?
Pardon the expression, but useless. We've tried that one as well. It was suggested, and it hasn't actually worked. It didn't work in New Zealand as well, in the published literature. So that's not effective. We still have not done experiments to look at the effects of oxytetracyclines or long-acting tetracyclines, like has been done for Theileria parva. That's the one piece of research that I'm probably not going to be able to do, but would be useful to do, I think, to see whether that has any effect on transmission. We know it has effect on transmission from the ticks, but we haven't been able to determine whether it has any effect on the development of the parasite in the animal, in the host.
Thanks, David.
Yep.
Next one's a comment from Phil. His understanding of the 28-day tick claim of Dectomax is that it has a residual activity of around a week, and then the other 21 days is the time taken for an adult tick to become noticeable again.
This is actual work done against cattle tick, and what they did was the standard thing for persistence is that you keep putting a new batch of ticks on at weekly or fortnightly intervals. And they found that if you really go through the paper and sort of and dissect it out, they found that they could actually still kill around 100% of the larvae at 28 days. So that's all I'm going on there, because obviously that tick can stay on for about 3 to 5 weeks. But if you can kill the larvae within 48 hours that are coming on 28 days after the application, then that's a good claim for, or a good analogy for bush tick. After that, they hadn't got anything that convinced me, but 28 days certainly they did. Again, it's off-label. And again, the injection worked a hell of a lot better than the pour-on, the topical.
Thanks, David. Next question is for you, Matt. Is the Northern Tablelands considered an endemic area?
Yeah, the LLS vets up in the Northern Tablelands did some great studies up there, and they found Theileria on almost all of the farms they went to, and there was a sort of theory that, you know, east of the highway it was endemic, so you know, Walker and places like that, and then west of the highway, once you got out, you know, beyond the other side of the New England Highway, they weren't there. But that's no longer the case. There does seem to be a lot of Theileria either side of the New England Highway in the Northern Tablelands.
But it's pretty much subclinical, isn't it?
Yeah, they don't seem to have the higher doses that we have, that you know, people see in the coast.
So it's either vectored by a tick out there, which is not as good a vector as bush tick, or that it's getting transferred, what I call mechanically, by some sort of mechanical means, which is only the blood stages. And we know from our studies that the transfer of blood stages doesn't actually cause clinical disease. And so it's why I actually mentioned that the cycle through the tick seems to be the sexual reproduction maintains the virulence of Ikeda and Chitose.
Thanks, David. Thanks, Matt.
It's sort of an evolving scenario, because we don't know what the cattle movements are, nor the seasonal variation in the ticks, whether they get carried with floods, or whether the kangaroos are having a great old feast and moving further west, or just how this is actually moving between seeing clinical cases and just seeing animals coming up PCR positive.
Thanks, David and Matt. Next question, I think I'll direct to you, David. Can sheep and goats act as intermediate hosts also? You mentioned kangaroos were susceptible.
I know that sheep can actually have parasites. Whether they're actually sufficiently high enough numbers of parasites to infect the bush tick, I'm just not sure. I haven't got any data on that.
Yeah,
but
there was some good work. Yeah, good work done in New Zealand on that, where they did show exactly that, David. But yeah, no one's claiming that they're a major part of the transmission cycle.
Next question's for you, David. Ali says, thanks, David, a very informative and interesting talk. And the question is about vaccine development. And from your experience, what do you think are some of the key challenges to developing an effective vaccine against Theileria?
The first is understanding the protective immune response, which for parva is quite easy, because the major component causing disease is actually the stage in the lymphocyte, the white blood cells themselves. And so we know all about how to develop that. And they've also got a very successful vaccine from Stabilate, which is the infect and treat one with oxytetracycline. The infective stage, or the most the disease-causing stage for Theileria orientalis, is this stage in the red blood cells, and that's a difficult one to work with. And probably it's a prevention is better than cure. We've got a long way to go, I think, to work out how to produce an effective vaccine against Theileria orientalis. My view, and there's a little bit in advance of where I'm up to, is that if I can work out what happens, how the carrier state actually protects against seasonal tick challenge, that'll be a very interesting one to do. What we have done is had some animals that have come in as carriers, and we've actually put ticks on them, and they have not developed any high level parasitemia at all. They've been significantly reduced compared to animals which are totally naive and susceptible. So certainly that carrier state does actually protect against the levels of parasitism we see after tick challenge under circumstances where the animals are totally susceptible.
Thanks, David. I'm quite sure you could talk in response to that question for the rest of the evening, so we'll move you on. Could insecticidal e-tags be effective for ticks? Well,
that's where the sort of the anecdotal evidence comes in for python tags. I mean, I got the stuff from the literature search from Queensland, where they've been used against Ixodes holocyclus paralysis tick. And okay, they're effective against buffalo fly. And so, you know, it was just like they did in Queensland for Ixodes holocyclus. We gave it a go with bush tick. We don't have any idea of how many bush ticks or whether it was effective against the tick itself, but it certainly, the only readout that we had was the fact that the parasite load was lower in the animals that had the tag. So I'm presuming ipsi facto that that the circumstantial evidence was it decreased the number of ticks or slowed down the infection rate.
Thanks, David. Matt, a question for you. Are you aware whether a poron deltamethrin will control bush ticks off-label?
Yeah, and again, off-label warning means we've sort of got an ethical obligation not to talk about off-label stuff. But the direct answer is probably, and for the same reasons that the spray-on works. You know, you're getting the chemical on the animal. Just be aware, when you spray an animal, you're actually applying chemical to a lot more part of the body than when you use a poron, and that could be a key part of it. I know when we're trying to clear up lice on cattle, that you know, applying a little bit of the poron to the pole of the head and the switch of the tail allows the animal then to spread the chemical to a lot more places than when you're just going straight down the back line. So that's a little tip. You know, there may be better results if you're using a poron compared to a spray. Try and achieve good application to those parts of the body that are most at risk of getting ticks.
Yeah, I think you probably have to also keep in mind whether you've got a knockdown or whether you've got any persistence, because these ticks are only on, bush ticks only on for about a week, and then it drops off and you get a new lot coming on. So if you just got knockdown, it's becoming pretty nonsensical to keep on pouring it on to control them. If you've got persistence like you have maybe with 14 to 28 days, then you've got a much greater chance of trying to make an effect on the tick population.
Thanks, David and Matt. We just have a couple of questions to round off the Q&A today, which are back to location. And we actually have a Northern Tablelands District vet online who just wanted to update you, which is fantastic. So thanks for letting us know that they're still getting clinical cases up here in the Northern Tablelands, and the last clinical case was in Inverell in January. So thanks very much for adding that to our conversation. It's great to have eyes on the ground then. So the next question, Matt, I'll give it to you for now. I've heard that the southeast of South Australia has had a few cases. How concerned should we be down here? And is the changing climate going to mean more cases may occur?
Yeah, good point, Kate. I was down in South Australia just recently, and as part of my homework, I looked at the summer rainfall for the southeast, and a lot more summer rain than in a typical year. And of course, successive La Niñas mean that it goes from being an unsuitable climate to, a semi-suitable climate for these ticks. So there's a good chance that, this is a time when you will get a higher risk than when you have, typical hot, dry summer conditions in South Australia. The cases that have been reported have come from around Millicent, and so we know that they were from cattle imported from other areas. There is a good chance, though, if cattle were brought in that had a lot of ticks on them and the ticks dropped off, they could survive and infest other animals and give them the infection, and of course cause death and disease subsequent to that. So yes, I do think you should be quite vigilant with your biosecurity and treat animals that you bring in.
Yep.
Thanks, Matt and David. That brings us to the end of question time. So thank you very much, both of you, for joining us today on the presentation.